afternoon, dear audience. So we are on one of the last sessions in this great forum, and I think this is one of the most exciting sessions because we are going to listen on our young scientists, those who will be the game changers in the future, I believe. So it's so important to listen on them, to support them, but also ask some good questions to trigger some ideas. Right, today the session is led by Riekstina, Professor Riekstina from University of Latvia.
And Professor Zanda Daneberga from Riga Stradins University.
Yeah. So we will be chairing this session. I believe that each of presenters have 10 minutes time to do the presentation, and we would welcome 1-2 questions after each speech. And if we will have still some time in the end of the session, because we are very restricted on the time, then we can do a panel discussion here, if we will have time for that. Okay, yeah. Let's start.
I think so, yeah. And I think after your talk, you return back, because then you will be able to follow other presentations. Yeah, okay. So I would like to invite the first speaker of today, young researcher session, and that is Olesja Rogoza, and PhD student from Riga Stradins University, with the title of the talk, Prediction of Breast Cancer Cell Fate Through BH3 Profiling. So please, the floor is yours.
Okay, you can hear me well? Okay. So as it was previously stated, yes, my name is Olesja and it's a pleasure to be here. And today I will talk about how can we predict breast cancer cell fate and use it to our advantage. Triple-negative breast cancer is a breast cancer subtype that is characterized by the lack of expression of 3 hormone receptors. It represents about 15% of all breast cancer cases and carries the worst prognosis. TNBC is an aggressive and heterogeneous disease, and unlike other clinical subtypes of breast cancer, this type of breast cancer lacks targeted therapies. And additionally, this type of cancer has high recurrence rate and strong resistance to therapies after the relapse. And despite the advances of treatment, for TNBC, the outcomes for many patients remain suboptimal, and due to various death evasion mechanisms cells employed to survive. And one of the factors contributing to this tumor heterogeneity and tumor cell survival is hypoxia, or in other words, oxygen deprivation. And while generally hypoxia is bad news for most cells and tissues, cancer cells, on the other hand, can survive and thrive in these conditions. And hypoxia has been linked to increased invasiveness, malignant progression, development of protective niches, and, as I previously stated, death evasion mechanisms that contribute to therapy resistance. And despite a pretty complicated title, our idea of our project is very simple. We look at triple-negative breast cancer cells under normoxia and hypoxia, with and without treatment, and then we evaluate how these cells differ and which cells survive. And therefore, the big question is, how can we destroy all of these cancerous cells that have survived? But before we think of a way how can we destroy all these cells, we need to understand how cell dies in the first place. Therefore, cell death is a fundamental process for all organisms, which is complex and multifaceted. And apoptosis is programmed cell death, which is initiated by signals either from outside of the cell or within the cell. Nevertheless, both outer and inner pathways result in the same outcome. Cell dies through but destructive enzymes known as caspases, and these caspases in the inner pathway are activated by cytochrome C, which is found in our powerhouses of the cell, mitochondria. And if there are holes in mitochondria, cytochrome C is released, it activates caspases, and cell dies. And scientifically, this formation of holes is called mitochondrial outer membrane permeabilization, or MOMP for short, but we will keep it simple. So how these holes are formed in the first place? These holes are formed in mitochondria when two proteins back and back interact and create a complex. And unless you're a cheesemaker aiming for the perfect Swiss style of the cheese, these holes generally are not desirable. Consequently, cell uses different strategies to avoid this formation of holes, and one of the way is by downregulating or inhibiting pro-apoptotic or pro-survival proteins, and the other way, or the strategy, is by upregulating anti-apoptotic or pro-survival proteins. Regardless of the strategy cell employs, the outcome is the same. There is no formation of holes and no cytochrome release, therefore cell survives. And apoptotic death is not something sudden. Cell is preparing for die end, and this concept is known as apoptotic priming. You can think of apoptotic priming something like a cliff. Some cells are closer to the edge than the others, and those closer to the edge we call highly primed because they're more likely to die, and those further away from the edge we call unprimed cells because they are not ready to die, at least yet. And it's important to measure this priming of the cells and understand the molecular basis behind it. And in this regard, Professor Aleksejs' lab developed a technique called BH3 profiling, which basically measures cells' readiness to die. And by knowing this balance between anti-apoptotic proteins and pro-apoptotic proteins and the specific binding between both protein classes, we can determine how ready the cell is to die and which anti-apoptotic strategy cell uses to survive. And the workflow for BH3 profiling is relatively simple. We plate cells in plates, seed cells in plates. Then we expose these cells to various specific BH3 peptides. Then we fix the cells to preserve the response to this peptide, and stain cells with DAPI, which stains nuclei, and cytochrome C, which you can see in this picture. Cells stained with cytochrome C are those who are unprimed or living and have retained cytochrome C inside. Then we proceed with imaging, image processing, and data analysis. And the first step of our project is to determine how primed the cell is initially under normal conditions, normoxia, and then see if these anti-apoptotic strategies cell uses persist under hypoxic conditions. And therefore, our preliminary results from normoxia experiments indicate that cell line HCC1143 likely is dependent on anti-apoptotic protein MCL1 because MS1 protein, which is pro-apoptotic protein, inhibits its functions. And this inhibition results in pore formation and release of cytochrome C. And in turn, cell line HCC1937 is likely dependent on either BSL2 or BSLW, and we can determine it as well in later stages using different peptides. And actually, this is my last slide, and I really want to acknowledge my fantastic colleagues and especially my mentor, my PI, Aleksejs. Thank you.
Thank you. We can take one or two questions. Yes, please. Yeah, there I can see the hand up.
Have you tried treating them with the MCL1 BH3 mimetic like AMG 176 to confirm your findings?
We actually treated HCC 1937 with venetoclax, and we saw that it's not dependent on BCL2, actually. So
And
there was no cytochrome
Did you try
release.
the MCL1 inhibitor AMG?
Not yet,
176?
not yet,
Yeah.
but it's in later stages of our project. Of course, we will try different mimetics, not only mimetics, but also chemotherapy drugs on cells.
Thanks very much.
Thank you. One more question? Yeah, maybe I can ask. Yeah, how did you choose the cell lines for experiment?
Since I joined the group only in later stages, it was the fantastic work of Aleksejs and the other colleagues, but these cell lines are one of the most popular cell lines used in triple-negative breast cancer research, commercially available.
Thank you. So thanks. I think we thank you for the wonderful presentation and good luck with your future work.
So now we invite to the floor next speaker, which is Ilze, a PhD student from University of Latvia, and she will be talking about influence of the acute myeloid leukemia microenvironment on the outcome of the therapy. So please, floor is yours.
Hello, I'm Ilze, and as mentioned, today I will talk about acute myeloid leukemia and more of the microenvironment model systems and development and innate immune cell role in therapy resistance. What is acute myeloid leukemia? It's a hematological disease with high lethality. It's a heterogeneous clonal disease, and it arises from a rare population of malignant stem cells from the bone marrow. If untreated, the lethal outcome in worst-case scenarios comes within weeks. And these specific types of AML are rare in general population and averages only 1% of all cancer cases, but it accounts for almost a third of the leukemia cases in adult population. And poor information on this is that the therapy resistance is really high, especially in elderly patients, where a 5-year survival rate is only 5 to 10%. Since AML is a very versatile disease, it's actually a task to choose right components when we're trying to model it in laboratory setting. And we need to know the big picture. What is the specific subtype of the cells of our interest? Do we need to choose? Oh, do we need to? Oh, yeah. Yeah. Do we need to choose a specific subtype, or we are looking on a broader picture of this disease? For example, THP1 is the most common and most cited cell line in AML research, but it represents only a small percentage of actual cases in real life. And the question is, could this be a good representative model when we are searching for new therapies and comparing to the patient samples? Maybe, maybe not. Since AML is a versatile disease, with different several subtypes, it is challenging to find the best approach to split the system up, to make it easy transferable into laboratory setting and model it, and in turn make these models relevant for patient sample analysis. So today I will try to introduce you to the basic components of AML and these techniques, how we model it in a laboratory, and give some bits and pieces of my PhD project. Tumor is not a single cell entity. It's a diverse organism. And this microenvironment consists of several different cell types, even in liquid tumors. And this environment can act as a protector of the tumor cells or act against it. And this shift in microenvironment is actually a hot topic to research. When we look at the innate immune cells that are inside this microenvironment, we can observe that there are two types of macrophages. Oh, wrong button. Macrophages acting in place. And it is known that when the microenvironment is overloaded with tryptophan metabolism byproducts like EDO1 and kynurenine, these macrophages switch their phenotype to more cancer protective phenotype. And thus exploring this relationship between macrophages and the cancer cells is really crucial for understanding the therapy resistance when it comes to AML. There are several approaches when researching and modeling the tumor microenvironment in vitro, and my take on this is a bottom-up approach, which means we start with the 2D monocultures and layer the system to 3D cultures and work our way up to make an organ-on-chip model. My goal of the project is to create acute myeloid leukemia microenvironment on a chip. And to succeed, there, of course, are steps. First, you need to know what elements are there in your model system and how they function individually. Then you can layer up the system and combine them with different cells. And lastly, the chip system allows us to conveniently model AML as an organ. So the first step of my PhD thesis phase is monitor macrophage polarization and how they change surface and secreted markers upon phenotype change, and how different cell lines act upon polarization, and if the selected marker detection panel can be applied to a versatile group of patient sample analysis. And then I model co-cultures, putting together tumor cells with healthy monocytes to monitor how the environment influences shift to more tumor protective environment, and if changing these parameters can make a difference in therapy resistance. As I mentioned before, there are two types of macrophages in microenvironment. M1 are more pro-inflammatory and anti-tumoral. M2 macrophages are more protective and pro-tumoral. And these forms can interchange. And this shift is called polarization. If we look at the methods of how to mimic this shift and cell polarization in vitro, there are some hurdles. For example, literature is divided on the right protocol, how to do it. And the big question is what happens when what are the outcomes when you use several different AML cell lines instead of the most popular one. And we have found that we can overcome this. Please play the movies. Somebody?
If you move on, it will start playing.
No? Oh, yes. So we use live cell imaging. And with live cell imaging, Oh, yes. We can monitor in real time different cell lines and see the morphological changes in real time. And live cell imaging can aid in determining the correct time points and parameters for optimal cell polarization. And here we can clearly see the differences between two AML cell lines and their morphological responses to the same polarizing conditions. As an endpoint analysis, imaging flow cytometry shows that we can definitely see the polarization effect on cells, and there is difference when we look at the markers expressed on healthy cells and malignant cell lines. But still we need to finalize the panel so we can use it to the patient samples, because they are really heterogeneous. So the take-home message is that there is a lack of synchronity of protocols and approaches when it comes to AML research in vitro, and different AML cell lines express different levels of markers. Thus, of course, this panel development for patient samples is an ongoing process. And the next steps for our research is to develop on-chip system to model AML in a more organ-like setting. And I would like to thank for the funding and my team for opportunity to do this work. Thanks.
Any questions from the audience?
Yeah.
Yes, just maybe wait for the microphone. First Ines, and then in the end. First, yeah, here. Yeah, please. And then you
Will
can
be
pass
faster.
microphone.
Yes. Thank you. It was a very interesting presentation. My question would be like, what is your expected readout? How are you going to detect the resistance?
At the moment,
Like live dead cells, morphology, or like what, like you were mentioning, because the aim is like to measure the resistance. Can you?
the aim is to measure resistance, but at the moment we are as well tinkering about BH3 profiling as well. But since the goal is to make it on a chip, it would be chip-suitable readouts that are more in line of flow cytometry, mass spectrometry, and cytokine secretions.
Yeah, and we had a question. Yeah.
Is it on? It's on. I have a very not specifically to your system. I have a question about one slide that you showed about this macrophage activation by tryptophan metabolites.
Yeah.
Can you describe briefly again, is this something specific that happens only localized places, or because in our research in the gut we see that it's there's a strong dysregulation in different disease phenotypes of kynurenine pathways that induce disease essentially. So it's a slightly
it's not limited to ML. I would think so, but what we are working is because it is shown in patient samples that this kynurenine overload is predicts poorer outcomes for patients. We are looking into this pathway to see how it affects this microenvironment and this immune cell polarization in particular.
Yeah, because I'm asking because there might be then a connection between, let's say, different types of diseases through this tryptophan pathway.
General mechanism of disease, yes.
All right, thank you.
And maybe I can very quickly ask one small question. I know it's all in the cell culturing and microenvironment, but me as the person with the genetic background, I would ask, do you have any plans to look at epigenetic changes during that transition which you described?
At the moment, not, but it is a possibility that we will grow our protocol spectrum also in some genetic field.
Good. Thank you. Good luck with that. Thank you. Okay, so the next presentation is from Evita Rostoka, who is the postdoctoral researcher at University of Latvia, and will be giving a speech on the topic determination therapeutic efficacy by PDSC, on-chip microfluidic system. Please, floor is yours.
Yes, it's working. So, hello, dear colleagues. Today I'm proud to tell you about our latest achievement, which was done here in Faculty of Medicine and Life Science of University of Latvia, under leadership of the tenured Professor Una Riekstina, and I jumped in this study a year ago, and I jumped not only in the study a year ago, but in oncology. So I consider myself as a very young scientist today. So, and yes, and today I would like to speak about pancreatic ductal adenocarcinoma, which is one of the most aggressive cancers commonly diagnosed in advanced stage. So, considering general aging of society, obesity, and epidemic of type 2 diabetes and other factors, so it's a PDAC incidents and PDAC-related deaths are expected to double in the next 10 years. So it's a very huge problem, I think, and it's, for me it's personal, because my parents died from this disease, so it's very, yeah, you can hear by my voice. So yes, and diagnosis of PDAC is difficult, and treatment often is ineffective. So there are growing demands for therapies, methods, I don't know, analysis, devices, and so on, to diagnose this cancer and treat it also. And thanks to Vadim's Parfeye's, organoids came into our laboratory, and we decided to use them, to use this technique to make an organ on chip. Yes, and the device, the chip, came from Institute of Solid-State Physics, from Robert Rimsha, and it is a very interesting device, if you can say so, with two channels, on the top one, and on the bottom other, and the porous membrane in between. And we know that PDAC is a very dense tumor, and this membrane simulates this density. And also we have inside this chip the flow in both channels. So it mimics more physiology, the situation in a cancer. We hope so. And yes, and what we did, so I mentioned before that we have this, Vadim Parfeyev, this organoid guru in our lab, and he teach us how to make these organoids from patient samples. And samples was from Austrumslimnīca, it's Eastern Hospital, in collaboration with Andres Cholkins. And we got the samples from hospital and we made, I think, more than 10 cell lines with these organoids. Okay, so what we do next? Of course, we characterize those samples, and we do it with Reinas Melngrove, also from Austrumu Slimnīca Eastern Hospital. And of course, we did this targeted DNA sequencing for pathogenic mutations, as we can see here. Yes, and also we looked for markers, and yes, we found this specific pancreatic progenitors and tumor markers. So yes, we got the sample. We got that tissue sample. And thanks to our very hard-working, beautiful PhD student, Karina Golova, yes, she did the work, and she made this organ on chip, which lives more than 50 days, 50. Here you can see in this picture that we had this PDAC tumor channel on the top and HUVEC channel that mimics blood vessel on the bottom, and there was separate flows in them. And after that, we got this outflow. So, and it looked something like that in our laboratory. It was very interesting to see how Karina managed with these tubes and this, I don't know, syringe and an incubator thing. So it was, thank you, Karina. It was an art. So, yes. And now I want to show you how it was under microscope. And there's few things, a lot of things we have, but I have to mention few things. So, can I go back? So, in the beginning, we also did that experiment on the plate, of course. And you can see here and here that we have also in both cell lines, HUVEC cells and in tumor lines. So we can see that morphologically it's very, I hope you can see, difference how look the cells under microscope. And then I just want to show you how beautiful it was under microscope, these channels. Yeah, day 15, day 25, and day 50. So you can see the density grows of the cells. But here we can see both channels, you know, in combined. And yeah, and here comes the interesting part, I think. We got these tumor-like cells in outflow. So maybe it was some kind of metastatic cell. I don't know. We don't know, but could be. And the other part, we did also that work. We changed the flow rate in the channels and measured what happens. And we saw that the permeability of this porous membrane decreases when that flow increased. So, yeah, it was very interesting for us. Okay, and the next slide. Yes, of course, we wanted, oh, sorry, I do not speak in the microphone, sorry. And yes, of course, we checked for that cell viability in a chip, and we measured with 3 different markers, LGH, it's here. It's so-called apoptotic marker, biomarker, so-called find me, fractal key, and of course caspase 3. And we did it in a chip and in ML, and how you can see here, the results was similar, very similar. So yes, we had this, I cannot say apoptosis, I can say that cells died more in this PDAC culture. So I can say, both in a well, in a chip, in a both. Yeah, And yes, we got this outflow that I mentioned before. And what we did, very simple thing, I think, but it was very interesting. We found in literature these PDSA biomarkers, and only one, CA 90.9, are used now in the clinic for diagnostic reasons, and others are just some kind of candidates. And what we found very interesting, we did the job in a chip, in both chips, we had 2 chips, and in a well. And how you can see here, for example, for TIMP1, red one is for PDSC channel, that level of this TIMP was up in a PDSC channel, in both chips, but in the well, we found it only in a well, in a medium from Helvijs cells. Okay, the next, okay, this I can understand the next one, but in with MIT1 and AXHL molecules, we have the very similar results. But with CI 90.9, opposite. So it was very, very interesting for us. Why it's so, we don't know. But we can speculate that it could be some kind of, you can imagine 2 channels in porous membrane with cell layers, and so those biomarkers somehow go through, or those cell lines, that tumor and HUVEC, some kind affected each other and do something with each other. We don't know what, but we have this result right now. Yeah, so I'm not too fast.
It's okay.
Yeah, so about conclusions, the main one, we did it, I think. So we established a personalized PDSC chip using, so this patient-derived organoids and HUVECs that mimic the vascular barrier and tumor interaction in an OOC system. So I think we did. But we also showed how cells are changing with flow and without flow. We found that something happens with those biomarkers, and so on and so on. So we have a lot of results, but we have a lot of questions also. And yes, so I think we really created this PDAC chip as a platform for future studies, for I don't know, pharmacological studies, for microenvironment studies, and so on. So yes, and if you want to learn in detail what we did, or not, so you can, yes, we published it, I don't know, 3 weeks ago, a month ago, recently. Yes, and you can go there and read. So if you want to ask ask questions, go there. Sorry. Okay, they are sent here, not all, but most of our team, and it's our building, this in University of Latvia. Thank you very much.
Thank you. I guess we have time only for one question because it was a bit over the time, but yeah, there's one question in the front here.
Don't get angry. My question was about the PDOs, the patient-derived organoids, and you were mentioning something about the flow and the influence of the flow on that. As I understand, it's like how many different flow rates did you check, and did it also affect the formation of the organoids, and also how long time it took for the organoids to... I understand they were formed in the channel.
Yeah.
Yeah. So
the organoids, no, we seeded them.
So you made organoids outside and then you seed it in the
You
channel.
know that you can say that girl. She can tell you in details everything. Yes,
Yeah.
but sorry.
But about this flow rate, how did it affected the viability of the PDOs? And also, I understand there were different flow rates for the endothelial cells and a different flow rate for the PDOs, or it was the same?
It was the same flow, but it was 3 flows. I think it's one. It was. I don't remember that. The numbers was 1, 2, and 4. But if Karina can assist me and tell in what.
Yeah, we checked.
Sorry, because it's not.
Make sure that the cells are not detached of it, and we chose the one 2 microliters per minute. So yeah, I don't.
So yes.
Okay, thank you. And maybe from my side, one very quick question, because you described that the primary patient-derived cells were characterized by somatic variants. Did you check those variants after you grew the organoids and how it's changed?
Yes.
Was it changed or was it more or less the same?
It was the same.
Okay,
and
that's good.
I think it was several passage
Yeah.
after. Yes, we checked.
Okay,
Yes, it was the
that's
same.
very good. Yeah, thank you. Thank you.
Okay, thank you. Well done. Okay, and we proceed with the next speaker. And I would like to invite Helvijs Niedra. Helvijs is a PhD student from Latvian Biomedical Research and Study Center. And his talk is going to be about digital spatial profiling of RNA in tumor cells in alpha SMA positive stromal cells of pancreatic neuroendocrine tumors. So, okay, the floor is yours. Welcome.
Hello. Okay, mic works very good. Thank you for the introduction and thank you for attending this seminar. And yeah, today I will talk about digital spatial profiling in pancreatic neuroendocrine tumors. So neuroendocrine tumors, while they are not as interesting as PDAC, they're still an interesting tumor because they're highly heterogeneous. Neuroendocrine tumors themselves are highly heterogeneous as they can arise in a lot of parts of body, and what really makes them neuroendocrine is that they have to arise from neuroendocrine cells, and main markers of neuroendocrine differentiation are synaptophysin and chromogranin. And this is uniformly expressed in all neuroendocrine tumors. And specifically, pancreatic neuroendocrine tumors are relatively rare but deadly, and they originate from islet cells in pancreas. And while their incidence is 1 in 100,000 annually on a global scale, there's a thing that most tumors, or 32 to 73% of cases, especially when it comes to these non-functioning tumors, they are diagnosed at metastatic stage. So that is what makes them lethal. And histologically, pancreatic neuroendocrine tumors are well-differentiated tumors, and to somewhat alleviate the classification, they are classified into 3 grades based on Ki-76 and mitotic rate. This is not ideal, but it's the best we got, and upcoming classifications are expected to include molecular markers such as ATRX and DAX, and there's also some publications about PDX1 and ARX. However, these markers are, well, tumor cell markers. What about microenvironment? It's a hot topic that's been studied a lot lately, as it's a significant component to any tumor's development. And one of the important parts of the microenvironment are the stromal cells, not only the immune cells, but also stromal cells. And they can promote tumorigenesis in various ways by, for example, carrying out the immune suppression, making tumor immune cold, or promoting metastasis by remodeling the extracellular matrix. And IHC-based studies have shown that upregulation of alpha-smooth muscle actin, or alpha-SMA in short, in tumor stromal compartments does correlate with worse clinical prognosis. And therefore, the aim of this study was to characterize gene expression profiles on RNA level in these alpha SMA expressing stromal cells and tumor cells across all 3 PMNET grades. And to do that, design of the study is quite simple. We had 8 pancreatic neuroendocrine tumor patients, 3 grade 1, 4 grade 2, and 1 grade 3. And we got FFP samples from them, and they were placed on slides and treated with antibodies for alpha SMA and simnotiphysin. Alpha SMA marks these alpha SMA expressing cells, and simnotiphysin is specific for this neuroendocrine tumor. And then these samples were also treated with the HTCA panel for geomics, which contains RNA binding probes for 1,800 known tumor and tumor microenvironment genes. The interesting thing about these probes is that they contain photocleavable oligos, and using this instrument, digital spatial profiler, we can find specific regions of interest, and then, for example, decide that we want to release these oligos only from alpha SMA expressing cells, and in that way we can pretty much profile the gene expression information from a cell that expresses either simnotiphysin or alpha SMA. And in total, 104 segments were profiled, sequenced, and the results. One of the first things I was interested in is understanding what kind of genes are characteristic in these alpha SMA expressing cells. And to do that, I selected a region in tumor that's rich in these cells and segmented it. And once I got these AOIs, areas of illumination, as it's called in this methodology, I compared them against other types of areas of illumination or AOIs, some that contain information from islet cells, tumor cells, and pancreatic acinar cells, in order to identify genes that overlap between all these comparisons. And the overlap, why did I try to look for overlapping genes is because, well, if they're upregulated uniformly in like all 3 comparisons, that means these genes probably are specific to the alpha SMA expressing cells. And once I did that, next step was to understand how do these genes function together by performing string enrichment analysis to understand their interaction on protein level, and of course carry out overrepresentation analysis to understand what kind of pathways are overrepresented by these genes. And it was completely expected that there will be pathways related to extracellular matrix organization and so on. But what's interesting, we also see signaling by PDGF, and MET activates PTK. And the same is also for the genes in grade 2 tumors. That was the first one for grade 1. The same is for grade 2, except we also see a signaling by Notch overrepresented in these genes. And yes, similar is in grade 3 tumor as well, as we can see MET, Notch, and PDGF signaling. Why did I focus on these 3 signaling pathways is that in recent studies they have been proposed as one of the ways how tumor may interact with the not microenvironment, but specifically stromal cells, cancer-associated fibroblasts, through these signaling pathways, especially Notch signaling and PDGF. Next, I also compared these alpha SMA stromal cell AOIs across different tumor grades, because it's already been established that there will be differences in gene expression in tumor cells from different grades of tumors, because, for example, grade 3 tumor cell has way higher proliferative capacity than grade 1. And naturally, I got curious and thought, what about stromal cells? Yeah, so I carried out comparison like this. And one thing I also did was so-called reverse deconvolution analysis, where you take the cell type deconvolution data and use that to further understand whether the genes that are differentially expressed between these comparisons are differentially expressed because they're differentially regulated in these cells, or because it just happened that in grade 3 tumors, in these segments I profiled, there just happened to be more immune cells or tumor cells I just couldn't really filter out. It's not like these cells are mostly stromal cells. No, there's also a little bit of mix of tumor and immune cells. And so if there are differences in cell type abundances between segments I compare, they will show up as differentially expressed genes. But using this approach, I can understand which genes really correlate with the cell type abundance and filter them out. And after doing that filtering and carrying out functional enrichment analysis, again, I see that there is also overrepresentation of the notch signaling and MET signaling and PDGF once again. And that is it about stroma. Last thing I want to talk about is that I also profiled the tumor cells themselves. And as I mentioned before, PMNETs arise from islet cells. So it was only natural that idea came in my mind, why not compare tumor against islets? Specifically because then I would be able to look more closely what has changed on RNA level in tumor, rather than, compare tumor against whole pancreas, and, the pancreas mostly is exocrine, and if I would be basically comparing endocrine cell against exocrine, and of course there will be differences. But this time I'm comparing the tumor against the cells of origin. And yeah, there are interesting results as overrepresented pathways in the differentially expressed gene lists are related to established oncogenic signaling pathways, such as MAPK, VEGF, which is discussed frequently in PMNETs, and also PI3K signaling. And one thing is, what I observed is that also there were 5 genes that were consistently downregulated in all 3 grades. And one of them, CD99, is already an established marker in Ewing's sarcoma. But in PMNETs, it hasn't really been studied, except for one study in 2004 that showed, it was an IHC-based study that showed that the loss of CD99 correlates with worse prognosis in these tumors. It's interesting that this marker hasn't really hasn't been studied further in PMNETs. And so the conclusions are that the use of DSP allowed to identify some potential core genes in stromal and tumor cells, and overexpressed genes and associated signaling pathways in stroma may indicate that there could be a crosstalk between tumor and stroma involving MET and PDGF and notch signaling. Of course, this needs to be validated using actual models. And yes, as PMNETs advance in grade, the gene expression profiles not only change within tumor, but to some extent also in stroma. That is it, and I would like to thank my PI, Evita and Latvian Biomedical Research and Study Center. And I recently managed to publish this study in Journal Molecular Oncology. If anyone's interested, here's the QR code.
Thank you. So you did it. You did it.
Yeah.
Okay. So questions, please, if any, are there? Yeah. Yes, please.
So I'm not so much familiar with this type of tumor. And so you say they arise from islets. So in islets there are different types of cells. Beta cells probably the most common. So the PMNETs can arise from both, right? From alpha cells and beta cells. Can you somehow deconvolute those in your analysis, or do you pursue that at all, or is it something too complicated to assess?
That's a good question. It came in my mind. The problem was finding a good data set, a single cell data set from which I could derive matrix for a cell type deconvolution. And also, since I used the panel that contained 1,800 genes, it's not whole transcriptome, the resolution may not be good enough to distinguish beta cells or alpha cells. But to say from what cells the PMNETs arise, there was a very good methylation study that showed that they arise from beta and alpha cells. And you can, using the methylation profiles, you can actually group them in beta cell type tumors and alpha cell type tumors. Yes, and it would be very beneficial, of course, if I could further deconvolute from what kind of cells they arose.
Mm-hmm. Yes, please, one more. Yeah.
Thank you. It was a very interesting presentation. Looking at the practical part of this of these results, like for example, this RBP4, which was like extremely downregulated in tumor cells versus islets. I think in one of the last slides, it was like very like wow. Would you suggest it as a biomarker? And if you would suggest it as a biomarker, how you would expect it to be measured in a clinical setting?
Yeah. How would I expect it to? Well, one way how to validate biomarkers, for example, the CD99, in a clinical setting, one way would be if they are not secretome proteins. One way would be just doing IHC study and see if these markers correlate with clinical parameters. But I don't want to be mistaken. RBP4 is retinol binding protein. I don't want to be mistaken, but somehow in my memory I think it's one of the secretome proteins. And if it is, it could also even be checked in blood.
Okay. I see
That was my aim, actually.
Okay. Oh, excellent, excellent. All right. Okay. We'd like to thank you once more, for presentation. It's a great presentation. Thank you. And I would like to invite our next speaker. That is Agnese Brokane, PhD student from Latvian Biomedical Research and Study Center. And Agnese is going to talk about the effects of cancer-derived extracellular vesicles on tumor microenvironment, the functional significance and potential use in precision medicine. So the floor is yours.
Yeah, so hello. It's an absolute honor to be here at this forum for the first year, and I would just like to share with you our research. Me and my colleagues at the Latvian Biomedical Research and Study Center have been working on for the past 2 years on extracellular vesicle roles in cancer communication and their potential use in precision medicine. So small introduction into EVs. They are small particles that are enclosed in a lipid bilayer membrane and that are secreted by almost all types of cells in the body and are also present in all biofluids. They are stable in the extracellular environment and can transfer information from their parent cell to distant cells throughout the body. Now, enclosed in EVs, first of all, on the surface of EVs, you can find various molecules such as tetraspanins, which are characteristic of their parent cells, and other membrane proteins. And enclosed in EVs, there are RNA and DNA molecules, lipids, proteins, and other metabolites. So currently, various studies have shown that all cells within the tumor microenvironment actually communicate through EVs. And in the case of colorectal cancer, it's been shown that cancer cell EVs promote tumor growth and chemotherapy resistance, whereas cancer-associated fibroblast EVs support tumor survival, cell migration, invasion, and cancer cell formation. Furthermore, there are also have been some potential biomarkers identified in blood plasma EVs that contain these diagnostic and prognostic markers. Now, it is known that EVs, along with tumor cells, can enter the patient's bloodstream, where their contents can be taken up by distal recipient cells in the body, and they can induce pathological changes in them. Now, we and others have actually studied the molecular cargo of EVs in hopes to identify EV-enclosed biomarkers to use as diagnostic and/or prognostic tools. And now, although some promising candidates have definitely been identified, none have, or very few of them have reached clinical validation. So we reasoned that an alternative to this identification of individual biomarkers would be to look at EVs as complex entities and to see their combined effects on the tumor microenvironment cells. So we asked the question, is it possible to detect these cancer EV-induced effects in an in vitro system and to use them in cancer diagnostics, monitoring, or prognostics? And to answer this question, so the first thing to do was to obtain a large amount of EVs, and to do this, a colorectal cancer cell line was cultivated in a bioreactor. EVs were isolated from colorectal cancer cell supernatant, cancer patient plasma EVs, as well as healthy donor EVs, and characterized accordingly. So we cultured this metastatic colorectal cancer cell line in the bioreactor pictured above, isolating EVs from the cell supernatant for a total yield of around 2 to the times 10 to the power of 12 EVs. And microscopy images showed their characteristic cup-shaped morphology and presence of EV markers, as well as purity of cellular debris, was confirmed by a Western blot. So the next thing to do is we wanted to see which types of cells present in the tumor microenvironment actually efficiently take up EVs, and 5 recipient cell types, including fibroblasts, monocytes, cancer cells, and also macrophages, were treated with EVs, and their uptake was assessed by flow cytometry and fluorescence microscopy. Oh, sorry. So we observed that fibroblasts, macrophages, and cancer cells efficiently took up EVs, both cancer cell and plasma EVs, although monocytes, well, not so much. So next, we wanted to see, we performed RNA transcriptome sequencing analysis on those cells that did in fact efficiently take up EVs, and we wanted to compare the changes in EV-treated cells versus non-treated cells. We saw that the strongest response to EVs was in HS68 fibroblasts, and from upregulated genes with a fold change larger than 1.35, we identified 8 genes uniquely induced by EVs and 45 shared with the protein co-isolated fractions. And from these EV-induced genes, most were mainly related to cell metabolism, angiogenesis, cell motility and migration, cell proliferation, adhesion, and differentiation. So the next thing to do was to validate these findings, and we selected candidates for validation based on their increased expression in cells that were treated with EVs compared to the negative control, increased expression in cells treated with EVs compared to the protein fraction, and also functional relation to cancer development and its processes. We selected in total 8 genes who we thought best fit our criteria, and the functions were also mainly related to cell metabolism, cell cycle regulation, and interactions with the extracellular matrix. So the first thing we did was we confirmed, we tested these candidates on HS68 cell RNA that we actually used for the sequencing experiment, and we did confirm that everything was correct, and the expression level of these genes was in fact elevated in cells treated with our cancer EVs, especially for the genes MMP10, 11, and STC1. And then we took these genes and we wanted to see if whether the expression of these 4 genes was somehow directly related to the concentration of EVs that were added to the cells. And we indeed saw a correlation between EV concentration and these gene levels, and this was also on a repeated experiment from cells of a different passage. So we also managed to validate these findings. And then we performed survival analysis on these genes, and between all colorectal cancer subtypes, a high expression of these genes was in fact correlated with poor overall patient survival. So then, having seen all these effects, cancer cell EV effects on our fibroblasts, we wanted to see if we would see a similar effect if we added EVs that were isolated from colorectal cancer patient blood plasma, and if we could somehow use this information in cancer diagnostics. So we performed a sequential sequencing experiment using colorectal cancer patient plasma EVs and healthy donor EVs. And now, if we compared these two groups, we saw a number of differentially expressed genes, and we were specifically looking for genes that were upregulated in cells treated with cancer patient EVs compared to healthy donor EVs, and also the negative control. So this time, most of these genes whose expression was changed by the addition of cancer EVs, cancer patient plasma EVs, was related to proliferation, angiogenesis, and metabolism. And at this point, we really wanted to validate our findings in an independent patient cohort. So we selected 4 candidate genes which were upregulated in cells treated with cancer patient plasma EVs compared to the healthy controls, and whose function was also somehow related to the cancer-associated fibroblast phenotype. And we also added our 4 most promising candidates from the previous experiments. And well, so based on our current data, the good news is that all of these genes do in fact seem to be induced by EVs. However, maybe their hoped diagnostic potential does seem to be a bit lackluster at the moment, with only 1 gene showing a close to significant elevation in cancer cell EV treated cells. But another question which we are currently working on is their prognostic potential, and the next step would be to see whether there exists a correlation between gene expression and cancer aggressiveness. So at the end, we concluded that our fibroblasts, macrophages, and cancer cells did effectively take up cancer cell EVs and blood plasma EVs. Cancer cell and patient EVs did induce gene expression changes in our fibroblasts related to various cell processes such as metabolism, molecular signaling pathways, proliferation, and others. EV-induced gene MMP1011, RGS9, and STC1 expression levels in our fibroblasts were dependent on the added cancer EV concentration and were related to poor overall survival. But most importantly, we believe we showed proof of principle that we were able to detect cancer-derived EV effects in an in vitro. However, we did realize that there are multiple external factors that affected the cellular response to EVs, and right now identifying them is crucial for developing new diagnostic tools and actually using our results further. So thank you very much. I would like to thank everybody that helped in this project, in the data acquisition and moral support, and most of all, I would like to thank my supervisor, Professor Aleksejs.
Thank you. We support you with applause. It was a great presentation. And questions, welcome. Yes, I see one hand there and then one there.
Thanks so much. So when you add the EVs to your stroma cells and you see that nice change in signaling, do they actually protect the colorectal cancer? Does it have a phenotypic effect, the change in the stroma cells? Can they protect the tumor cells more, or do they? Did you do the co-culture?
I have to be honest with you. I personally did not do that in this project. I believe a colleague of mine did do a similar experiment, but it was, I think it was a different system, so I can't really comment on the results. But yeah, but well, from the sequencing results, we did see that, you know, those EV-induced genes were somehow related to the cancer-associated fibroblast phenotype, so maybe.
Thanks very much.
Thank you. Next question, please.
It's actually a very, very similar question to Trina. I think it is like super cool to see like how the other cells are taking up the EVs. You're like kind of focusing on the genes, but I'm a biologist, and I would be like, how does these EVs change the cell biology? Like, for example, a normal fibroblast, did it make it more cancerous fibroblasts, or you said that it made the difference in gene expression of signaling pathways? It's kind of like very generic thing. Like, did it kind of move this cell towards kind of a little bit more cancerous or a little bit more abnormal cell type? And especially when we look at the macrophages as well, that was kind of very interesting, I think, area as well, to look at how the EVs, cancer EVs, affect these cell biology.
Yeah, so your question was similar, I guess, to the previous one? Just how
these cancer cells, cancer cell EVs affected the biology of the normal cells, fibroblasts, and the macrophages. Like the functions, did they proliferate faster? Do they, I don't know, do they have some more cancerous phenotype or something?
Well, I can say yes, because I've read it a lot in other publications. Now, in this project, we were, of course, focused solely on sort of identifying these biomarker candidates. So we didn't quite look at how we didn't perform any assays that would directly tell us how it would impact the fibroblast proliferation. As for macrophages, we actually ended up excluding macrophages from the sequencing experiment, as we weren't sure if their EV uptake was specific or non-specific, if they were just eating the IVs and taking them everywhere into the lysosomes, and then and in the lysosomes everything was just degraded and expelled out. So we actually did exclude macrophages in the end, as we were, I guess, too afraid to work with them. But for fibroblasts, obviously, it's it would be wonderful to get an opportunity to actually perform these extra experiments, but they have been previously reported in other studies. So yes.
Thank you. Yeah, wonderful work. Good luck in future.
So now we come to the last presentation of today, which is going to be presented by Aleksejs Fedulovs, who is a PhD student at University of Latvia, and he will be going to talk about individual risk factors for glycemic variability and personalized approach for type 1 diabetes patients, including AE solutions. Please.
Yeah, thank you for kind introduction, and good day, colleagues. Today I would like to present the findings on our study, the clustering of type 1 diabetes patients by CGM profiles, and the higher insulin resistance and the risk of MAFLD assessed by surrogate markers in patients with poorly controlled glycemia. This research addresses an important growing concern in management of type 1 diabetes, which is the rising prevalence of double diabetes and the metabolic dysfunction associated fatty liver disease, or MAFLD, in this patient group. The prevalence of double diabetes and metabolic dysfunction associated fatty liver disease is increasing among the patients with type 1 diabetes. In addition to being an independent cardiovascular risk factor frequently associated with metabolic syndrome, MAFLD might lead to deterioration of glycemic control. The regulation between glycemia management and the liver outcomes is little studied in type 1 diabetes, and there are no data on how continuous glucose monitoring outcomes and MAFLD risks correlate between each other. The aim of the study was to investigate the differences in MAFLD risk markers, insulin resistance, and metabolic syndrome prevalence between clusters of type 1 diabetes patients based on CGM profiles. The study was cross-sectional and included 75 patients with diabetes type 1, and the patients were divided in 2 clusters based on the CGM data, such as average glucose, coefficients of variance, glucose management indicator, and time above the range, time in range, time below the range, and low glucose events, using the hierarchical clustering with WARD method and Euclidean distance. The CGM was done by diagnostic freestyle Liber Pro IQ sensor and were worn for 14 days. Time in range was defined as glycemia between 4 and 10 millimoles per liter. The risk of muffled was assessed by fatty liver index and the hepatic steatosis index, which was calculated by those formulas. And the prevalence of metabolic syndrome was assessed according to the NCAP criteria, and insulin sensitivity was assessed by estimated glucose disposal rate formula. So the hierarchical clustering is an unsupervised machine learning technique used to group similar data points based on a distant metric. It produces tree-like structure called the dendrogram, and that represents how the data points are merged into clusters. As we can see, our findings reveal 2 distinct clusters, cluster 1 consisting of 28 patients with poorer CGM outcome, and cluster 2 consisting of 47 patients with clinically better CGM profile. These 2 clusters did not differ significantly in age, gender, diabetes duration, and the significant difference emerged in their glycemic control and muffled risk markers. Yeah, as we can see on this slide, the differences were really huge between those 2 groups, and we can see that the cluster 1 had shorter time in range and fewer time below the range events. This cluster also had higher GMI and more time above the target range, as well as higher HbA1c that we will see in the next one. The patients also in the cluster 1 exhibited higher liver enzymes such as alanine transaminase and aspartate aminotransferase, indicating potential liver stress. Also, both the hepatic steatosis index and fatty liver index were higher in cluster 1. And also the estimated glucose disposal rate were lower in cluster 1, as we can see, and the prevalence of metabolic syndrome was not statistically significant, but still higher in the cluster 1. As for the study highlights the association between the poor glycemic control measured by CGM profiles and the increased risk of metabolic dysfunction associated fatty liver disease in type 1 diabetes patients. Similarly, a recent study also investigated the metabolic determinants of muffled in individuals with type 1 diabetes that found that poor glycemic control, defined as HbA1c below the 7%, above the 7%, doubled the risk of developing of muffled. The patients in cluster 1 who had worse CGM profiles and had higher prevalence of metabolic syndrome exhibited significantly lower estimated glucose disposal rates, indicating a higher insulin resistance in this group. Those findings align with the other research also where a similar association between insulin resistance and as measured by estimated glucose disposal rate and poor CGM profiles was observed in people with type 1 diabetes. This patient, the patients with type 1 diabetes with poor glycemic control should be more actively screened for muffled using non-invasive markers like fatty liver index and hepatic steatosis index. Early detection of the muffled could allow for more targeted interventions to prevent the further liver damage. And also the clustering approach using the CGM data provides a novel way to stratify type 1 diabetes patients by their risk for complications such as muffled by identifying those with higher risks by clustering. And of course the clinician can prioritize those patients for more intense interventions such as dietary modifications or exercise recommendations, or also the insulin therapy modifications. As for the conclusion, for now, our study discovered the links between glucose daily profiles assessed by CGM and muffled and insulin resistance markers in patients with type 1 diabetes using a clustering approach. But for our further plans, we will check the also inflammation markers also in both clusters, and then also get new conclusions more. I would like to thank also the head of this research work, Ilze, and also would like to thank our statistician who did all the hard math stuff in this study, Leonora Pahirko. Thank you for your attention.
Questions from the audience? I can see one here in the front.
It's me. I'm sorry, but to be honest, I understand what's the MMP, but I do not understand. Can you explain for me what is the time below in or above the average range? I mean, I'm like,
of course. Like the time above the range is the time that patients had higher than the normal blood sugar level. So, for example, in our study, it's above the 10 millimoles per liter, and usually this standard is set by the sensor by itself. It's like a default value. If above the 10 millimoles per liter, then it counts as a time above the target. So the person wears CGM sensor for 14 days, and from that 14 days, 60% of the time he had blood sugar level above the 10 millimoles per liter. Same go for below the target, and same go in target. Of course, those clusters, if I could go back, I don't know. No. Those clusters both are bad. Like there is no like good cluster and bad cluster. The whole group is with pretty poor CGM outcomes. So, but as we can see, there is like group with very poor results and the poor results, basically. So there is no good results, unfortunately.
Any other questions from the audience? Okay, maybe I can use the situation and ask one. More on the terminology point of view. So your presentation said it's individual risk factors. Would you consider it as the risk factors or more like a biomarkers for indicating for this variability of the glucose level?
You mean like if those they are individual because we need to like get those clusters or get those hepatic steatosis index values, fatty liver values, estimated glucose disposal rate. We need to know a lot of stuff about one particular person. That's why maybe they are called like they are directed for one person, because at least person need to use this method as we described. We need to know a lot about one person, so it starts with at least with the anthropometric measurements, and he should warn the sensor for 14 days. So it's a pretty like individual approach in this case.
My question wasn't not about individual, but risk factor or biomarker for indicating the changes in this glycemic variability.
Maybe can you rephrase that question?
So, like, if you think on the physiological point of view, would you see this as the risk factor in general because it's not linked with the disease, or this is like biomarker which is physiologically linked with the disease?
It's linked with disease. That's probably the next step we need to do. We need to explore the inflammatory markers that are also involved in the muffled process. So that's our next step that I mentioned. So we will check the CRP at least. CRP already saw that it is correlating in cluster 1, but for other markers we still need to do more work. So.
Yeah, and have you calculated the sensitivity of this kind of markers, how well it can predict?
I will not answer this question, sorry.
Thank you. Yeah, thank you very much.
I would like to invite all speakers in front to sit here in the panel, because it's a really, really excellent opportunity for you to exercise how it's going to be in the future more and more during your PhD and academic career, and so that we all can see these wonderful, brave speakers and give a huge applause to the work they've done. And yeah, we have, I believe, 4 minutes for a question session. So if there's any question that you still want to ask, so now it's the time to do it. So questions welcome.
Maybe while audience is thinking, I can ask
Sure.
the same question to every one of you, as we are on the network forum of the personalized medicine, and your topics of your research is obviously in the field. So, but you are in some early stage or maybe a bit later stage of your research. So how you see how your discovery or research results in the future can give an impact to the personalized treatment in the patients in real medical care. And you are allowed to dream as well. So, which would be brave enough to start, please? Just go on.
Hi. I think if I can dream and create an organ-on-chip model, then I think it's the pinnacle of the personalized medicine when you can take one person's sample and make it an organ out of it and study anything you can imagine and help to relieve the treatment time and find new cures for disease. So I think it's my work is the first steps toward it.
Thank you.
Stone? No? Yes? Okay, maybe I can say about our method, which is called BH3 profiling. It's very direct. Direct for method and with direct benefit for precision medicine, because we can use it not only in solid cancers, we can use it in hemato-oncological cancers. We can see the direct effect not only in normoxic, physioxic, hypoxic conditions, which is very like a hallmark for solid cancers, this hypoxia, and we can use it with and without treatment, how specific treatments work or do not work for each patient based on this method.
Thank you. Who is the next?
Well, as for me, I think, well, obviously it would be great if we could finally develop an EV-based diagnostic method that actually works, you know, because, well, first of all, we would be very rich. So, you know, but second of all, I mean, I genuinely believe that there is something to extracellular vesicles, you know, and all of this data, like these decades that people have been working with them, there is definitely something there, you know, and if, I mean, obviously the biggest challenges right now are with EV, with protocol standardization and sort of, you know, the experiment reproducibility, as you saw. But I believe that if we, you know, if we could get past these challenges and develop, you know, a method where you could use these gene panels, you know, to diagnose cancer, obviously in liquid biopsies, which is also, you know, a non-invasive method that everybody wants to do, and yeah, and you know, and if we could also somehow alter these gene panels for certain maybe subsets of patients in the future, it you know, it would be great, and I would be rich, and my life would be great.
Yeah, it's working. Yeah. So for us in our study, as we saw, we already have those data. So the patients have the CGM outputs that are the clustering is based on, and also every other parameter is already available for each patient. So we just need to figure out how to make more use of those data, of this data. Yeah. Just in the future, I hope that there will be no bad cluster and very bad cluster, at least that one will be good. So that's why basically we need to make use of those data because they anyway are collected. So the patients are wearing those CGM sensors, and maybe in future with help of the AI, and we can improve parameters like detect the risks earlier for those patients, and we just need to develop the methods how to do that. So that's the basically the answer.
Thank you. Still Helvijs, and then question from the audience.
Okay, so I don't really know what to say, but philosophically speaking, cancer is extremely complex disease, and a lot of things have gone awry, and we can't really cure it with just omics, as there was a presentation, and my study is mostly omics. And yeah, I honestly just hope that my data is robust enough and it's good enough, and I've described it in a good enough way that it creates hypotheses for functional studies, so that can be tested in vitro, in vivo, and perhaps develop novel treatment strategies that can target microenvironment.
So yes, I am with Ilze. We are from the same laboratory, and yes, I will speak also about organ on chip, but I think that cancer is not the only disease what we have, and I work a lot with autoimmune diseases and diabetes, and I think we can help not only cancer patients with the system, but maybe also others.
Thank you. And the question from the audience?
Actually, it wasn't a question, it was a little bit a comment, because what I wanted to remind some young scientists that actually these methods that we are developing, the thing that we are working for the patients. And I'm sorry, this is not the way how we want to get rich. I'm so sorry, I know you were honestly answering this, but I wanted to remind you that actually the aim of this field is to help the patient, to help, as in one of the presentations we heard today, that we sometimes don't think about it, but I think Dr. Sieving talked about it. We never think about it, but sometimes we can get in this other side. Sometimes we can become the patients, yes? So we have to be very careful when we are, to understand, what's our motivation for what we are working for. Thank you.
Thank you. And I would like to conclude the session now. I think it was very productive. You learned a lot. And I wish all the best of success with your research. And thank you for the audience for very interesting questions and for this discussion about values as well. And yeah, I would like to conclude with giving a big applause again to all of us. And thank you to co-chair Zanda.